Drosophila Melanogaster. These have been studied for a long time. For a while, they weren't used, but then they were brought back into play because of genetic studies, because of further genetic studies and genetic manipulation. So in early stages, when we had experimental embryology, we used them, and then they weren't showing us as much as some of the other organisms. And then when we started getting into genetic manipulation, it was all about Drosophila, which is why they're used quite extensively as the invertebrate model for development. So they're perfect for genetic studies. There are homologs, which are genes that have the same function in Drosophila as they do in us, which is why when we study them here, we're studying ourselves as well. One of the biggest ones that we study here are the Hox genes, which play a big role in vertebrate development. Now, they don't have Hox genes in Drosophila. They're called homeotic selector genes, but they are homologous. They are homologs in terms of their function in patterning. So, very ideal for genetic studies. They have a very rapid life cycle. Do lots of breeding with them. They have 4 huge chromosomes. They call them polytene chromosomes. And one of the advantages of these polytene chromosomes is you know exactly where the genes are on these chromosomes. It's not like us having 46 chromosomes where you have to line them all up and put them in this order and find out like that. There are 4 huge chromosomes in the Drosophila where you just know exactly where these genes are found. And one of the biggest advantages of Drosophila is being able to manipulate their genetics and then see various mutations that come about. They're very easy to mutate and then see what happens. They're used quite extensively for genetic breeding studies as well as genetic manipulation studies. It's all about Drosophila, really. Now, the oocyte initially is surrounded by what we call nursing cells and follicle cells. They play a huge role in setting up those maternal components, the RNA that's necessary to begin the patterning of the Drosophila. We're going to talk about genes. So we haven't really talked about too many genes now. Now we are, because these genes have been so thoroughly studied, we know what role they play in the development of Drosophila. Remember that Drosophila have superficial cleavage and syncytial specification. So what's the difference? Again, superficial cleavage is where, because of the thickness of the yolk, as the cells undergo mitosis, as they're undergoing, and they skip the growth phase, they're just undergoing DNA replication, and then DNA synthesis, and then division, and then so on and so forth. They just go through those 2 cycles for several periods of time. Eventually, all the nuclei are pushed out to the perimeter, the superficial layer, where the middle pretty much has the yolk, and you see all these nuclei around the edge. Eventually, the cytoplasm will be pushed down and around each of these nuclei, creating individual cells. So superficial cleavage. Cleavage doesn't occur until all the nuclei are around the periphery of the cell, and then at a certain stage, the cytoplasm moves and sequesters around each one of these nuclei, and they become individual cells. Okay? The initial stages of cleavage, the first 12 occur very, very rapidly, every, I believe, 8 minutes or so. And then there comes a point where it slows down. About the 13th cell division, it starts slowing down. And one of the biggest things that happens there is, in these initial stages, it's all about the maternal RNA, where they're being translated into protein, and they're establishing these gradients. There comes a point where these gradients then start turning on the genomic material. They start transcribing proteins from these nuclei here to start creating even more gradients. One of the proteins that's responsible for that transition, kind of mid to late transition, is called SMOG. Yes, named after the dragon in The Hobbit. Okay, so SMOG is the protein. Now, what does it do? It actually sits, as it's being made, it starts sitting and preventing transcription or translation of the maternal RNA. So SMOG is one of those RNAs that's in there, and as translation gets initiated, as it builds up, then it eventually stops the translation of the maternal RNA, and then those proteins can only access the genomic DNA to start transcription and then translation. Okay, so cleavage, superficial cleavage, if you look at the cytoskeleton or the cytoskeleton and the cytoplasm here, all the nuclei get sequestered around the periphery. Eventually, these actin filaments will pull the cell membrane down and divide each of these cells off, and then you'll have all of these cellular blastoderm all around the periphery of the Drosophila. Okay? At that point, so much has already happened, and that's what we're going to discuss, is once the nuclei have been sequestered within their own cells, there are so many different patterns of morphogen gradients that get pulled into those individual nuclei, that is what initiates further gastrulation and specification. So it's necessary that these nuclei are not sequestered into their individual cells until those morphogen gradients are set up throughout the entire cytoplasm of the oocyte. So before their cleavage has occurred and sequestered each one of these, they're called a syncytial blastoderm because it's all just one cell. And all of the nuclei are within one common cytoplasm. Eventually, once they all get partitioned off, then they're called cellular blastoderm because they all have their individual single cells that will then start interacting with one another. So that mid-blastula transition, where the mitotic rate slows down, that's what SMOG helps out with, is it is involved right about that mid-blastula transition that slows down maternal RNA transcription, and you get increase of genomic RNA transcription going on. Now, in terms of gastrulation, gastrulation is, again, the movement of the cells. You get invagination, you get convergent extension and epiboly and whatnot. I'm not going to spend too much time on this, although we'll go over a few key aspects of it. One of the things is at the ventral region, you get invagination of cells that will eventually become the mesoderm. So the mesoderm forms in the ventral, very similar to the sea urchin in that the mesoderm is in the ventral region and then it pulls up inward and invaginates inward. The only difference is in sea urchins, it's ingression, and here it's actual invagination of those of that tissue. You can see it right here. So here in red, this is the ventral region of the Drosophila. The cells will invaginate and actually pinch off and form this almost tube that will become the mesoderm. The ectoderm and the neural tissue eventually will surround it, and the neural tissue will be in between the mesoderm and the ectoderm. That starts forming the nervous system. Eventually, these cells, these endoderm cells, will be completely engulfed through the gastrulation movement, as well as the germline cells. There's a few cells here, these called pull cells, that will go with the endoderm cells that will kind of be pulled in with them and then become part, and that will form the sex cells, so the sperm and the egg, respectively, based upon the sex of the Drosophila. So here's looking at the ventral region. Here's what we call the ventral furrow, where the ventral cells are invaginating and eventually will pinch off inward. And here's the closing off of the ventral furrow, where the cells are now internally. One of the disadvantages you can see here is we can't see internally. They're not clear. The cells aren't clear like the other two, like sea urchins and C. elegans. So we can see kind of what's going on the outside, but we can't watch all the cells. We've done studies where they've cut them open, and that's why we know what's happening internally. But it's a little harder than just being able to watch the cells do their thing without having to manipulate them at all. This region right here also invaginates. We call this the cephalic furrow, where it's going to separate the head region from the body region. And this is just another gastrulation process of invagination that separates those two regions. Here we're looking at the dorsal end. You can see the pole cells. These are the germline cells. that will eventually be pulled in as the endoderm undergoes what we call conversion extension, where the cells just undergo mitosis and they move and then they get pulled into the cell. Later on, you can actually see the segments starting to form. And we're going to show tomorrow all of the genes that are responsible for causing these gastrulation movements that will cause these segments to occur within the Drosophila. Eventually, you get the patterning of the legs and the wings. One of the things we've shown that we're going to get to as well tomorrow is these patternings have everything to do with the initial stages of these maternal components. And as they get set up, it turns on the homeotic selector genes, or the HUX genes, the HUX genes in us, homeotic selector in the Drosophila. And we've shown that if you cause different HUX genes to be expressed in different regions, you can actually get flies with 2 sets of wings. Or you can get arms where the eyes should be, and things of that sort. Kind of crazy stuff. So we're going to go into all the genomic dynamics of how this is done. Most of what we study as far as axis formation in Drosophila is anterior-posterior polarity. That's the major thing we use Drosophila for. We do know some of the aspects of dorsal ventral patterning in the Drosophila, and we do know some aspects of left-right patterning. But we know the most about anterior-posterior patterning, and it is absolutely fascinating to show how it goes from the maternal components to the genomic components to the homeotic selector genes that eventually form each of the segments of the Drosophila. And we've seen direct correlations between key transcription factors and the eventual morphology of the things that come about from that. The nursing cells and the follicle cells will put mRNA of many different types, but the main ones we're going to look at are bicoid, hunchback, nanos, caudal, and torso. These are the 5 that are what we call the maternal effect genes. And we're called the maternal effect genes because they're put in by the maternal cells, the nursing cells and the follicle cells that get the ball rolling. Okay? This is a perfect example of why the cytoplasm is not distributed symmetrically, why it's all asymmetrically distributed, and where you can start the initial body plan of the organism. And most of what we're going to talk about today is the anterior-posterior axis. I am going to make quick mention of the dorsal ventral axis, and it's very easy, primarily determined by a protein called dorsal. You want to know where dorsal is actually expressed? In the ventral region. Yeah, whatever. So I don't know why they do that to us. But let's start with this. Bicoid and nanos. These are the 2 major ones. Now, bicoid is found in the anterior region of the oocyte. So unlike the C. elegans, where there is no anterior-posterior pole until fertilization begins, in the Drosophila, it's already predetermined, the anterior and the posterior end. In fact, there's only a tiny area in the posterior end of the oocyte that allows sperm to enter in. Sperm cannot enter into anywhere else in the oocyte. It can only enter into a small little region in the posterior end of the oocyte. Now, the flagellum on the Drosophila sperm is so long that by the time that the nuclei fuse together and the sperm is entering in, that tail essentially plugs up the hole to prevent any other polyspermy or any other sperm from being able to come in. So that's one of the main mechanisms of Drosophila to prevent polyspermy, is that the sperm's flagellum is so long that it just plugs up the hole. Okay, it's to prevent other sperm from entering into that region. It doesn't have some of the same effects. However, it does have a reaction even before fertilization begins, and this reaction causes some of this initiation process. So it's not necessarily the fertilization process that initiates these changes in the cytoplasm. It has more to do with other mechanisms. So bicoid is the mRNA that is then translated into protein. Now these form what we call morphogen gradients, which are just high concentrations of protein that are diffusing out from a high to a low concentration across the cytoplasm. So if you look, you can see that the bicoid mRNA is sequestered to the most anterior end of the oocyte, and as the protein is being translated from the RNA, it just diffuses out, and you form this morphogen gradient. High levels of bicoid in the anterior region, and it gets less and less as it moves more posteriorly through diffusion. On the opposite end, you have what we call nanos. Nanos messenger RNA, again, gets translated into nanos protein, and this diffuses as a morphogen gradient as well, from the most posterior end to the anterior. So eventually, you have high levels of bicoid on the anterior, high levels of nanos, and then it just slowly grades down, and that's why we call it a morphogen gradient. There's other proteins that are also being made. Hunchback and caudal are actually initially dispersed evenly throughout the entire cytoplasm of the oocyte. So they're everywhere. Hunchback and caudal start out everywhere. But as bicoid and nanos start being translated, bicoid inhibits caudal, and nanos inhibits hunchback. So it starts restricting hunchback to the more anterior end, and it restricts caudal. Remember, I used the words rostral and caudal. Restricts, if you think caudal and posterior, it restricts caudal to the more posterior end of that. Let me see if I can show a picture here. So here, if these are the levels, initially, hunchback and caudal are everywhere within the oocyte. But as bicoid and nanos are translated into protein, and you get these morphogen gradients, high levels in the anterior end of bicoid, high levels of nanos in the posterior end, then nanos or bicoid starts preventing caudal from being translated and made into a protein. So caudal becomes restricted to the posterior end. And the same thing is true. Nanos prevents hunchback from being translated, and then you only get hunchback in the more anterior region. So that's why these are what we call the anterior and posterior maternal effect genes. So bicoid and hunchback are the anterior maternal effect genes. They cause the oocyte to be the anterior pole or polarity, and nanos and caudal the posterior. Now, torso is not only in one place, it's actually found in both ends, in the most anterior and the most posterior end. They found that the combination of torso and bicoid is necessary for the formation of the head, and torso and nanos is necessary for the formation of the tail. So torso isn't in one place, it's actually in both extremities. So torso isn't really an anterior-posterior maternal effect gene, but it is necessary in both regions to define those terminal boundaries of the anterior and the posterior pole. So it plays a dual role. It plays as a single protein, it plays a dual role in combining with these maternal effect genes to form that, because they found that when they inhibited torso, you didn't form a head. And if they inhibited torso in the posterior region, you didn't form a tail. They needed that maternal effect gene in both regions. So that's what this is summarizing right here. Bicoid and hunchback in the most anterior region, nanos and caudal in the most posterior region, and torso in both the anterior and the posterior region. These are the 5 maternal effect genes that I will expect you to know and describe just like I did on how they work together. It's very easy as far as bicoid and nanos, but let me go over hunchback and caudal again. Hunchback and caudal are evenly distributed throughout the entire cytoplasm. But bicoid essentially downregulates or prevents caudal from being translated into protein. And nanos prevents hunchback from being translated into protein. So that's what restricts hunchback to the anterior region and what restricts caudal to the posterior region. Gap genes. Those are the next group of genes. Then comes the parallel genes and the segment polarity genes. So let's start with the gap genes. The gap genes are interesting. Now, the gap genes get turned on by the maternal effect genes. So, and some of these gap genes are also maternal effect genes. Well, really, in fact, just one, hunchback. So hunchback comes back again with a vengeance in the second stage of anterior-posterior patterning. So hunchback is really the only maternal effect gene that is also a gap gene. So you turn on some new genes through transcription and then translation through these pathways, through the bicoid being transcription factors for certain key genes, and nanos being transcription factor for other key genes, combined with hunchback, combined with caudal, and all of these factors. Okay? So here are some of the main ones. We've got cruple, NERPs, hunchback, and giant. We won't worry about tailless right now. These are the four main ones: cruple, NERPs, hunchback, and giant. Now, here is the relationship that I want you to understand. One of the things that tends to confuse people at first is they're like, wait, why is hunchback in the posterior end? Well, what this shows is where it's expressed. It doesn't actually tell you the levels of it. So in fact, over here, there are very low levels of hunchback. You don't have high levels of hunchback, even though hunchback is an anterior gene. But hunchback does get turned on by some of these other genes, and as a gap gene gets expressed in these regions. Now, there's a hierarchy in these genes. And what do I mean by hierarchy is certain genes repress other genes. So the hierarchy is as follows. Hunchback is the highest on the totem pole. It has the highest hierarchy, which means that in the presence of hunchback, it will prevent the expression of giant, NERPs, and cruple. So where you get these bands of hunchback being expressed, it prevents any of these other genes from being expressed in those regions. Now, giant is next on the list. So giant precedes or supersedes NERPs and cruple. And one of the interesting things about this patterning is when you get the combined effect of bicoid, nanos, hunchback, and caudal, when giant gets turned on, if hunchback isn't here, giant would be expressed right here. But since hunchback is here, giant is restricted to here. Over here, again, giant is only restricted to here because hunchback prevents giant from being expressed right there. So you start creating these band patterns that will form the segments and the pair rule genes and the segment polarity genes we're going to talk about. What's interesting is because these are still gradients, these are morphogen gradients still, that they repress the expression of certain genes right next to them. For example, giant will keep cruple restricted to a tiny little area right there. Because giant's expressed right here and right here, it only allows cruple to be expressed right here. And in terms of hunchback, it restricts NERPs to this band right here, because hunchback is expressed here and here, and due to this hierarchy, it creates this very specific gap gene expression pattern. And you don't have to understand much more than that, but these are the gap genes. This hierarchy is what I want you to understand. Wherever hunchback expression occurs, you can't get giant, NERPs, or cruple. Wherever giant expression is at, you can't get NERPs or cruple. And wherever NERPs expression is at, you don't get cruple. So cruple is the low man on the totem pole, so to speak. It gets restricted to the only area where you don't get any of these other gap genes being expressed. And they reinforce one another. These morphogen gradients keep themselves in very specific bands, because if cruple were to try to expand, then number one, NERPs prevents it, and number two, giant in this morphogen gradient keeps it restricted to this tiny little expression pattern. The reason why I mention this is because, again, this is just diffusion of these proteins. So the only thing that keeps these in these very restricted bands is the fact that other proteins, like giant, will repress gene expression of some of these other proteins. That's what keeps them in these very specific locations in the anterior-posterior patterning. And it took a lot of work for them to figure out this hierarchy. A lot of, misexpression and knockdown experiments and such. So the gap genes then lead to the next set of transcription factors, which we call the pair rule genes. Now, there are lots of pair rule genes. There's quite a few. The only ones that I really want to talk about are Fuchitsuratsu and even skipped. Those are some of the pair rule genes. Now, you can see that they form very discrete banding patterns, and they don't necessarily all overlap with one another. So Fuchitsuratsu and even skipped, the way that they're set up, they just have very distinct banding patterns, but they're not found necessarily in the exact same banding patterns. Even though there's about 7 bands There's a wide variety of other pair-ruled genes. And you start getting this overlap of all of these genes that are spaced out amongst the embryo. So that's all the pair-ruled genes that I'm going to talk about, Fuchiteratsu, or FTZ, and even skip. Now, the reason why I want to talk about those two pair-ruled genes is because these are the ones that make all the difference when it comes to the next group, which is the segment polarity genes. Now, at this point of the segment polarity genes, this is where the syncytial nuclei, or the syncytial blastoderm, become cellular blastoderm. Up until this point, all the nuclei have been exposed to the cytoplasm, and all of these morphogen gradients that have been established, and all of these proteins that are being set up in these very discrete banding patterns along the anterior-posterior axis, now the nuclei get enclosed in with the cell membrane, and with that, all of the necessary factors, transcription factors and such, get pulled into the cell as well. And that's what starts causing these cells to then take on very different fates and start interacting with one another. Up until this point, it's all been morphogen gradients. But now we're going to get into paracrine signaling from cell to cell. We didn't call it paracrine signaling initially because it's all one cell. There's no paracrine signaling because they're not multiple cells. Now, with the segment polarity genes, each of these cells, nuclei, have been enclosed in the cell membrane, and they'll start expressing various receptors and various proteins. So the segment polarity genes that I want to talk about should be very familiar to you. In Drosophila, it's engrailed and wingless, but their homologs are none other than sonic hedgehog and Wnt. These are, as you should know, sonic hedgehog pathway and the Wnt signal transduction pathway that we talked about. Here, we get reciprocal induction. So the cells that express Wnt are in areas that don't have any Fuchiteratsu or even skipped protein. They're in these gaps right between these segment polarity, or between these pair-ruled genes. That's why I brought those two up. And there's lots of other things going on here, but I'm trying to keep it as simple as I possibly can. So Fuchiteratsu and even skip. You can see these. This is kind of the banding expression pattern of them. Notice how they don't overlap necessarily with one another. But in these tiny little gaps in between them, that's where these cells have the necessary transcription factors that will turn on the Wnt protein. Okay? In other cells where you have high levels of, it's either even skipped or Fuchiteratsu. It happens in both. But at high levels of even skipped and Fuchiteratsu, you get sonic hedgehog, or in the case of Drosophila, we call it engrailed. So what happens is these cells that are producing the paracrine factor engrailed have a receptor, which is the frizzled receptor, which the Wnt, or wingless protein here, binds to. And all this does, this reciprocal induction, this cell makes hedgehog, sonic hedgehog, that turns on the Wnt protein here. And the Wnt protein, in turn, through the Wnt signal transduction pathway, turns on sonic hedgehog. So all these do is just maintain Wnt signaling and sonic hedgehog signaling, or in the case of Drosophila, engrailed and wingless. That is one of the biggest things we've shown or seen in the development that starts causing a lot of the gastrulation movements that are necessary to create the segments of the Drosophila. What ends up happening is these two become signaling centers, where Wnt just gets expressed in a gradient fashion to these adjacent cells, and sonic hedgehog gets expressed in a gradient fashion to these other cells, and that in turn will turn on various genes that are necessary for gastrulation and changes in the cell's differentiation. Okay, so the two segment polarity genes that I want you to know are engrailed and wingless, the homologs being hedgehog, sonic hedgehog, and Wnt. One more difficult hurdle to get over. The last group of genes. Up until this point, each group of genes has built upon the last. The maternal effect genes turn on the gap genes turn on the pair rule genes turn on the segment polarity genes. Well, the homeotic selector genes, or the HUX genes, don't get turned on by the segment polarity genes. They get turned on by the gap genes and the pair rule genes. They've shown that that's the case, that the gap genes and the pair rule genes are what cause the patterning of the homeotic selector genes. So that's the only exception to this stepwise process of anterior-posterior patterning. So let's look at this. Homeotic genes, or HUX genes in vertebrates. As I mentioned before, these are in the same order on the chromosomes as they are in their anterior to posterior expression. I mean, it's just fascinating to say the least. Same thing is true for the HUX genes. They're in the same expression pattern in anterior to posterior and in various regions in our body as they're found on in that. You don't have like HUX 10 here and then 7 there and then 9 there. You get HUX 12, 11, 10, 9, 8, 7, so on and so forth. It's just fascinating how that works. So, homeotic selector genes. They don't have the same names as we have. In fact, when we do the HUX genes, we just give them numbers, you know, in terms of which gene that they are. The homeotic selector genes, however, have very specific names. Some of the more prominent ones that you may hear are things like Antennapedia, ultrabithorax. Ultrabithorax is really one that's studied quite a bit, as they've done studies where if they remove ultrabithorax, then Antennapedia actually takes its place, and you get 2 sets of wings like that instead of 1. So they've shown that these HUX genes, or homeotic selector genes, are necessary for proper development of certain structures. If you delete the ultrabithorax gene and you get more of this gene going in this region, you get 2 wings, or 2 sets of wings instead of 1. Another thing is if you have expression of things such as Antennapedia, which would normally make these legs in the eye region, then you end up getting limbs in the eyes instead of the eyes themselves. So they've found that these homeotic selector genes play a critical role in further gastrulation and differentiation of key parts of the Drosophila for the whole body plan. Okay? One of the things, too, about these is there is a hierarchy on this as well, where each one of these, going from posterior to anterior, supersedes the other, which means the abdominal B gene will prevent abdominal A gene from being expressed in this region. It supersedes that gene, and so on and so forth as you go through here. So if you remove ultrabithorax, that's why Antennapedia will just take its place because it's not being inhibited by ultrabithorax. So you're seeing the same phenomena here as you saw with the gap genes, where there's a hierarchy of these genes where one can prevent the expression. When you make Antennapedia B or abdominal B expression here, you will not get expression of these other homeotic selector genes, and so on and so forth. Now, this expression pattern that you see right here, again, is solely responsible for which 2 groups of genes? Gap genes and pair rule genes. Good. And that's it. That's as simple as I can make it. I know when you're reading your book, it goes through so much. I told you that I'd mentioned briefly about the dorsal ventralization of Drosophila. It's really simple. We know a little bit about it, but the main thing that causes it is there's a protein called dorsal. Okay, they call it dorsal. That's initially found everywhere in the embryo. Well, in the initial stages, what happens is when you get certain proteins being turned on, dorsal is removed from combining with another protein, and it is restricted to the freaking ventral region. So dorsal is expressed in the ventral region of the Drosophila, and that's what creates the initial stages of gene patterning that create the dorsal ventral axis of the Drosophila.